Okay. Yep.
Well, good morning. Good morning. Welcome to John Lacey's exit seminar. John asked me to introduce him. I'm Carl Bradley, one of his co-advisors. Ardra Katru is the other co-advisor. John's working on his, defending his master's thesis today. A little bit about John. He was born in California, lived there until he was about 14 years old, and saw the light and moved to Southern Illinois, which is a great place. If you don't know me, that's where I'm from as well. So he moved to a small town, Benton, Illinois, home of the Benton Rangers in Southern Illinois. He graduated high school and decided that college life he wasn't quite ready for college life, so he went to go work for the Millwright Union. And he did that for a couple of years, working in the Midwest, and decided that that was the time, now that he was doing hard labor, that it was time to think about college. So he ended up going to Southern Illinois University, home of the Salukis, and he was a plant biology major there. But he worked for Dr. Jason Bond's program, who's in plant and soil sciences at Southern Illinois University. Dr. Jason Bond is a plant pathologist, so that's where John was first kind of introduced to plant pathology, and he worked in his program as an undergraduate intern. I met John. Dr. Bond actually sent me an email about John and said that he was interested in graduate school. And I met John sometime in 2016, I think, and then brought him on board the summer of 2016 to begin working out at Princeton that summer. And then he started his graduate program that fall, coming here on campus, and then coming to Princeton in summer of 2017, and is working on Trichoderma as a biological control in soybean to manage some soybean diseases. So with that, John, I'll turn it over to you.
Thank you. Hello, everybody. Thank you for joining me today at such an early hour to listen about some interesting research I was able to conduct while at the University of Kentucky and its satellite research station, UK-REC, which is located in Princeton, Kentucky. The goal of this research was to evaluate trichoderma isolates for the management of soybean diseases. Objective 1 was a greenhouse trial to evaluate the ability of trichoderma species to colonize fungicide-treated seed. Objective 2 evaluated trichoderma species' ability to manage Rhizoctonia root rot. And Objective 3 evaluated trichoderma's ability to manage the foliar pathogen, frog eye leaf spot. The worldwide production of soybean is an important source of food, protein, and oil for humanity. Soybeans can be grown in a variety of environments, even nitrogen-depleted soils. Most often, soybeans are grown in production systems that rotate with corn, wheat, and cotton. One benefit of adding soybeans to a crop rotation is the supplemental nitrogen that the crop provides to the soil. Production of soybeans are widely prevalent due to the cultivar selection and the adaptability of the protein from the plant. Management of field crop diseases can be achieved by a combination of strategies, including host resistance, application of a fungicide, or cultural practices. The most effective form of management is the use of resistant cultivars. However, in some instances, a resistant cultivar cannot be used. If this is the case, fungicides can be considered as an alternative option for management. One downside of fungicides are the loss of efficacy over time, correlated to the development of resistant pathogen subpopulations. In recent years, the biofungicide market share has increased exponentially, and this growth is expected to continue over the next 10 years. The initial discovery of trichoderma and its ability to prevent disease was attributed to rapid colonization of substrates, outcompeting pathogens for nutrients, mycoparasitism, and the production of antibiotics. As intriguing as the initial findings were, the scientific technology of the early 1900s lacked the ability to harness the true capabilities of trichoderma species. More recent research on trichoderma suggests that the reduced disease severity was not a result of the previously characterized mechanisms. Interestingly, the ability Trichoderma has for managing disease is attributed to an increase in plant vigor or the induction of plant defense pathways. Currently, the biological control organism with the largest market share is Trichoderma, with over 60% of commercial products containing the organism. Before delving into the first objective of evaluating the ability of Trichoderma species to colonize fungicide-treated seed, I'd like to touch on some background research to help give you context of why this experiment was performed. Initially, 58 biological control agents were isolated from soybean roots in the north-central regions. These were obtained from healthy plants that were in high disease severity locations. 14 were screened for their ability to control common seedling diseases, and 11 of those were selected to test for fungicide sensitivity. These were the treatments that were used in this study. 5 different products that are commonly used, each have a different number of active ingredients. These active ingredients are used in the combinations as a broader spectrum for control of seedling pathogens. One of the ways farmers try and manage seedling diseases is to use a fungicide-treated seed. If we want to implement Trichoderma into existing management strategies for controlling disease, its ability to colonize a fungicide-treated seed must first be determined. Pictured here are the 2 isolates exhibiting the lowest sensitivity to common fungicide seed treatments that were selected for the greenhouse assay. These were Trichoderma humatum, Trichoderma virens, and the negative control Bionectria ochrolea. The goal of objective 1 was to determine the ability of Trichoderma isolates to colonize a fungicide-treated seed. A total of 5 fungicide treatments were tested with 3 organisms. Treatments were assessed for plant growth benefits by recording plant height at 7, 14, 21, and 28 days. Systemic effects of the fungicide and biological control agent were recorded by determining phytotoxicity on the cotyledon leaves at 7 days. Plants were harvested after 28 days to record top weight, root weight, and root length. And a qPCR assay was developed to detect the presence of Trichoderma in the roots of soybean. Most fungal genomic studies use the internal transcribed spacer as an identification at the species level. However, it did not work for this study because the negative control of Bionectria was detected. So to kind of overcome this issue, a degenerate primer was designed based on the endochitinase region of Trichoderma, which is a 42 kilodalton protein, which is a gene region that regulates a 42 kilodalton protein that acts as a chitinase. The soybean actin gene was used as a housekeeping gene for comparing to the expression of the endochitinase fungal gene. The amplicon size was 150 base pairs. As you can see in this lane here, lane 1, that isolate had issues with actin expression, so I decided to disregard that in the analysis. Before going into the data objective of study 1, I want to quickly show the beneficial effects of pure culture fungi when added to a non-treated soybean seed, since all treatments were compared to the untreated check. I want to point out that the fungicide treatments that had significantly lower values than the non-treated control, but this is an expected response. There were no significant differences observed in top height with the pure culture isolates. No significant differences were observed for top weight with the pure isolates. No significant differences were observed for root weight with the pure isolates. However, there were significant differences observed for the T. humatum and Bionectria ochrolea isolates, applied to untreated soybean seed. Now I'm going to go into what the actual objective of the study was looking at, which is colonization of the root. Fungicide treatments did not allow this particular fungus to colonize the root. For T. hilmatum, the only treatment that was able to colonize was the treated control. Okay, so the non-treated control was a soybean seed with no fungicide treatment. The treated control was pure isolate added to a soybean seed that did not have a fungicide component. All of the Trichoderma virans treatments had a significantly greater amount of fungus in the root when compared to the non-treated control. As expected, the Bionectaria treatment was not detected in the qPCR analysis. While there is slight variation between plate runs, the T. virans treatment had the highest rates of colonization for the fungicide-treated seed. The T. hilmatum isolate was unable to colonize the fungicide-treated seed, and the viability of Trichoderma species when exposed to evergrowth energy is a promising finding and should be pursued more in depth. For future experiments, there are aspects of the assay to improve. One bottleneck to the data analysis was that each organism with the fungicide treatment required 70 wells on a reaction plate. Since a single plate only has 96 wells, you couldn't compare between treatments of organisms. Now I'll discuss objective 2, which is evaluating Trichoderma species to manage Rhizoctonia root rot. In soybeans, the symptoms of Rhizoctonia solani include seed rot, root rot, hypocotyl rot, and aerial blight. Effective management of Rhizoctonia root rot is difficult due to an extensive host range and its ability to form overwintering structures known as sclerotia. These structures allow the fungus to survive multiple growing seasons. The ability of Trichoderma isolates to manage Rhizoctonia root rot was assessed by using a combination of stand counts, vigor ratings, and plant promotion parameters measured at harvest. Stand counts were determined by counting the number of emerged plants within a plot, measured by the amount of seeds planted. Vigor ratings for each plot were taken using a Trimble Green Seeker scanner. This instrument has a camera that detects near red and far red light, and then comparisons are made between the two channels, which results in the normalized difference of vegetation index. At 28 days, top height, top weight, root length, and root weight were recorded. The field assays performed to determine Trichoderma species' ability to control Rhizoctonia solani are referred to as hill plots. The hill plot design is typically used to determine efficacy of a compound or seed trait on a small scale. Each plot was prepared by adding 5 grams of inoculum, which you see here in this slide. The inoculum was prepared by growing a culture of Rhizoctonia on sterilized red sorghum seed. A total of 10 soybeans were added to each plot. Then the addition of 100 milliliters of either water or biological control agent at 10 to the 9 concentration per mil. A total of 5 treatments were replicated 5 times. The untreated check and inoculated, T. hilmatum 1, T. hilmatum 2, and T. virans. The Rhizoctonia trials were conducted at the University of Kentucky Research and Education Center, located in Princeton, Kentucky. Each of the Rhizoctonia sites had different crop production histories. The Catlett tract on the left was previously cropped as soybean, and the Luttrell tract on the right was previously managed as tall fescue. Data was analyzed in SAS using a general linear model, and this incorporates several different statistical analysis, which include an ANOVA, an ANCOVA, a MANOVA, a MANCOVA, linear regression, t-test, and f-test. After the data is analyzed, it was tested for significance using the Fisher's protected least significant difference test. The main idea of an LSD is to compute the smallest significant difference between two means. If the p-value is lower than 0.10, then the treatment is considered significant. Significantly lower treatments for the Catlett tract stand count at 14 days were T-hematum 1 and T-hematum 2. One treatment was significantly lower at the Catlett tract. This was T-hematum. Significantly greater treatments for the Catlett tract stand count at 14 days were T-hematum 2 and T-virins. There were no statistically different treatments for the Catlett tract root length at 28 days. One treatment was significantly lower for the Catlett tract root length at 28 days, and this was T-hematum 1. At 14 days, we see the same result with the T-hematum 1 isolate. And root length at 28 days, the T-hematum 2 isolate was significantly lower. So my two leading hypotheses for the reduction in stand are the lack of compatibility of T-hematum with the soybean cultivar, or the fungus ability to utilize the seed as a substrate and increase growth. Now we're shifting to the foliar fungicide application in Objective 3, where Trichoderma species were evaluated for their ability to manage frog eye leaf spot. Cercospora sogina is a polycyclic pathogen of soybeans. The initial symptoms appear on the leaf, stem, or pods. Lesions begin as small, light brown circular spots that exhibit a chlorotic to tan center with dark brown margins. If the infected area of the leaf is greater than 50%, the leaves can die and wither. Additionally, high disease severity can reduce the oil and protein concentration of the seed by 6.9 and 5% respectively. A large portion of the population are resistant to the quinone outside inhibitors, which are a fungicide. Field trials were set up in 2 different locations. 5 treatments were replicated 4 times. It was set up using a randomized complete block design. The fungicide or biological control agents were applied at the R3 growth stage, and this is beginning pod development. The plots were assessed for disease severity 3 times in 2-week intervals from the R5 stage to R8. Yield and seed moisture were recorded at harvest, and plots were 4 rows wide with 76 centimeter row spacings and 6 meters long. The fungicides and potential biocontrol agents were applied to the foliar portion of soybeans at the R3 developmental stage using a CO2 backpack sprayer. If applied correctly, yield and seed quality can be improved from a fungicide. Both field trials were set up using a randomized complete block. There were 5 total treatments, which were replicated 4 times. Defaconazole and azoxystrobin are referred to as quadrus top. Disease severity was assessed as area under the degrees progress curve, which is a useful summary of disease intensity over time. 3 foliar ratings were recorded during a 6-week period and were assessed in 2-week intervals. For the assessment of disease severity in the soybean trial, a researcher will walk through a plot that's 20 feet long and sample 6 random trifoliates within that plot. At the catlet tract, the disease severity was high, over 80% at the time of application. The only treatment that was significantly lower in disease severity when compared to the non-treated control was difeconazole and azoxystrobin. None of the Trichoderma isolates had lower disease severity. Interestingly, when looking at yield, only the T-virins treatment had increased. At the Luttrell tract, the disease severity was much lower at the time of application and throughout the trial, roughly 50%. And the difeconazole and azoxystrobin with the T-virins treatments had AUDPC values that were significantly lower than the non-treated control. All the treatments at the catlet tract or at the Luttrell tract resulted in yields that were significantly higher when compared to the control. Conclusions. At the catlet tract, when compared to the non-treated control, the treatment of difeconazole and azoxystrobin was able to reduce disease severity, but did not increase yields. The lack of yield increase could be from the disease severity at the time of application and throughout the experiment. The ability of T-virins treatment to increase yield, but lack of reducing disease, could be attributed to the release of plant growth regulating compounds. Further research is needed to identify these mechanisms that are causing this response. Objective 1, conclusions. Evergoal Energy is a possible fungicide to be used in conjunction with T-virins. Objective 2, Trichoderma species tested were not able to prevent Rhizoctonia root rot from occurring in soybean. And for objective 3, the Trichoderma virens isolate is a possible biocontrol agent, although its mechanisms need to be properly characterized. Thank you. I'd like to acknowledge Dr. Bradley's lab. I really couldn't have done this research without Kelsey, Josh, Danilo, Connor, and Jared. Being halfway across the state, 3 hours across the state is really difficult to manage field trials, and they were there anytime I needed them, which is a really good feeling to have with your research team. Dr. Ardra Katru's lab, technicians Wendy and Cation, research analyst Shine, Van, Tim, Riying, Huazen, Jaichi, Erica, Tinyu. You're all great scientists. And Dr. Fakori's lab at Southern Illinois University, his postdoc, Ali Saror, was kind of guiding scientists initially, and then, yeah. Thank you. We have time for questions.